Yeah, good morning, students. So, this is a part 2 of chapter 1 of Pharmacognosy subject. It's about the development and scope application of pharmacognosy. So, my next slides will be talking about the importance or the development of pharmacognosy. And before that, I would like to give some kind of hints about the scope of pharmacognosy. It's an extensive, indiscriminate use of synthetically derived drugs and antibiotics have led to the precipitation of several adverse effects and development of resistance by the microorganisms to these drugs. Therefore, the focus has now shifted to the usage of drugs derived from natural sources. If you talk about the growth opportunity of pharmacognosy, it is an associated industry. Phytochemical associated industry is high due to two reasons. India, being a country with a different climatic zone, is an ideal location for cultivating wide range of medicinal plants. This is the first reason. There, the growth opportunity of pharmacognosy is there. Second one, the advent of newer and efficacious separation techniques like thin layer chromatography, high performance chromatography, aligned with some spectroscopical techniques which enabled for the isolation of a newer compound from the plants, and acts as a natural source of drugs. However, increased demand for plant-based drugs cannot be met with the available natural resources such as forest or wild sources. Therefore, large-scale cultivation is required. There, the pharmacognosist role is there. And when we talk about the export of drugs, there are drugs from India which is exported to other countries. Artemisinin, good antimalarial drug. Artemisia annua is the plant. Podophyllitoxin is a good anti-cancer drug. Podophyllum exandrum. Silymarin, it's a wonderful hepatoprotective drug from the natural source called the Silybum marianum. Like that, plenty of drugs are there which is exported from India. There, the cultivation areas, the productions areas involved in different sectors. There, the role of pharmacognosist is there. And modern techniques, if I talk about modern medicine, in spite of tremendous advances, has not been able to provide a complete cure, but provides only what we call symptomatic relief from diseases like, an example, hepatic disorders, Parkinsonism, obesity, alcoholism, rheumatic disorders, even for genetic diseases like and virus disease like AIDS and cancer. It won't cure completely. It will give a symptomatic relief only. However, the practitioners of traditional medicine have claimed to provide a complete cure of these diseases. Therefore, such claims should not be ignored but scientifically evaluated, since report screening of active constituents of the plant provide ample scope for discovering newer means of treating dreadful and incurable diseases. And this was I fetched from one of the book that I prescribed yesterday from the T. Sham. So by this scope aspect, I move on to my next slide. Types of drugs derived from plants. So there are some drugs are derived from plants. First one, it has its role, right? Here the pharmacognosy I'm talking is a scope you can consider, or a modern development you consider, or the application you consider. So types of drugs derived from plants plays its role. What is that? Herbal drugs derived from specific parts of the medicinal plants. It has many pharmacological uses and it can be used as a medicine. Compounds isolated from plant extract. From the part of the plant, medicinal plant, one can go for extractions. From the extracts, one can isolate a pure compound that I mentioned in the middle of the image, You can see the plant and its relevant structural analogs. Each one comes under the phytocompound. Each phytocompound possesses a particular segment of therapeutic activity. And the types of drugs derived from plants, which also includes nutraceuticals or functional fruits. Nutraceutical functional fruits are also available in the form of capsule, tablet and syrup, and sometimes it is in the form of granules. They are made from the vegetables or maybe from fruits and other medicinal plants. The second one, the scope, or I can say the involvement of the pharmacognosy it provides a value of natural products. What is that? Compounds from natural sources play four significant roles in modern medicine. 1. They provide a number of extremely useful drugs that are difficult, if not possible, to produce commercially by synthetic means. It means we are not able to derive or synthesize commercially in a synthetic mean. For that conditions, the naturally obtaining products can be used as a medicine. Natural sources also supply basic compounds that may be modified slightly to render them more effective or less toxic. So from the natural sources, one can isolate a compound. So the basic compound can be obtained and that can be tested. It may show less effective, or sometimes it may be a toxic. And by the help of the computational chemistry, other drug discovery process, the structure can be little modified. By that, they can evaluate whether it possesses a more effectiveness, thereby it reduced any any toxicity level. This and all can be achieved from the starting material of the natural product. Points to be continued in another 2 slides also. For another third one, from the natural products, what we get, the utility as prototypes or models for synthetic drugs possessing physiological activities similar to the originals. For an example, the first structure is about salicylic acid. It is a precursor for the synthesis for the aspirin. Salicylic acid, with the help of the acetic anhydride, and it will, with the heating condition, it gives the aspirin. And you know what is mentioned? This salicylic acid prototype can help for the synthesis of newer drugs. You see the third structure, ibuprofen. But the salicylic acid original precursor structure is obtained from the plant source as a phytocompound. So that was as a lead for the synthesis of the newer synthetic molecules. You know what is the use of ibuprofen and the aspirin. Like, some natural products contain compounds that demonstrate little or no activity themselves, but which can be modified by chemical or biological methods to produce potent drugs, not easily obtained by other methods. For an example, bactacin 3. It is obtained from the Bacta plant, Texabacta plant, a anti-cancer plant. But the 3 is not much potent. So the idea from the bactacin 3, people have derived or modified its structure, and they synthesized a compound called as Taxol. It is a compound originally obtained from the bark of the Pacific yew tree, which has been found to inhibit the growth of certain cancers. So now Taxol is modified from bactacin 3, and bactacin 3 modified to the Taxol, it possesses a good anti-cancer property. And come to the production of natural drugs product. So pharmacognosy involved in the production of natural drugs products. How can we produce natural drugs? One, by a collection. Widely we can, by the wild source we can collect, first point. Second, we can do cultivation commercially, that can be collected, harvested, go for drying and packaging, storage it, like ginseng, ginkgo biloba, peppermint. The ginseng, ginkgo and all are good immune booster drugs. Now it's working like anything in the market. And by this, both are conventional. Now I'm talking about little deeper, recombinant DNA technology or genetically engineered drugs. It means the gene which is the gene present in the particular plant can be tied up with the other gene, thereby that plant material become resistant from any other environmental circumstances, like any bacterial or viral attack comes, it won't affect that particular plant. This is one way. Because of the genetical modification of the particular plant, the chemical content which is forming in the plant, there may be chances of increasing, or there may be chances of forming a newer compound. So thereby we'll get a new lead molecule from the plant. So this kind of research is going on extensively in worldwide, called as recombinant DNA technology or genetically engineered drugs, we can say. And cell culture techniques called as micropropagation. So this is the continuation of part 2 lecture only. Due to certain circumstances I have not recorded from this slide. So let me to continue. I think I talked about the micropropagation. Micropropagation is the growing of a plant cell in an artificial supported media to develop a new plant that may be a virus resistant and environmental resistant plant. And microbial transformation, you have the evidence that the antibiotics what we used is from the microbiological sources only, and there may be a biological products also produced from the natural product. It can be prepared from bloods of animals. Wonderful example is bovine serum. The bovine serum is wonderful for the therapeutical segment, and from there one can go for the isolation of thrombin. A thrombin possesses a good blood coagulation property which can be used during the surgical time. So the production of natural products way is collection, cultivation, recombinant DNA technology, and cell culture, and biologicals prepared from the blood of animals. The role of natural products in drug discovery. So how these natural products involve in drug discovery? One is the drug discovery can be happen by combinatorial chemistry. It comprises chemical synthetic methods that make it possible to prepare a large number, 10 to thousands or even millions of compounds in a single process can be achieved. These compound libraries can be made as mixtures, sets of individual compounds, or chemical structures generated by computer software. It means what we do in laboratory as a synthesis of one compound, like that we can do the stimulation by the help of some software. A group of series of possibility compounds will be shown in the picture, and that can save in the library. Each one can be subjected for the testing. And high throughput screening of natural product is a drug discovery process that allows automated testing of large number of chemical and biological compounds for a specific biological target. This high throughput screening in most of the journals, if you say HTSP, they mention like that, or HTSS. What is mean? The derived compound or stimulated synthesized compound in combinatorial chemistry, that each compound may be a micromolecule called as ligand. We can call as ligand. In our organ, in particular tissue, have certain some type of protein. That protein will be located in the receptor area. For example, for the cancer, in the cancer tissues, the protein, some proteins are involved. That protein we can identify with the help of protein data bank. That identified protein can be can be stimulated with the help of software for the interaction studies. So our synthesized compound or stimulated structure with the receptor of the particular protein, and the affinity and the docking score gives you the information how effective the drug may be. And the third one, combinatorial biosynthesis. It is in biotechnology. Combinatorial biology is the creation of large number of compounds, usually proteins or peptides, through technologies such as phase display. Here the peptides and protein based drugs can be synthesized. And not last, on this drug discovery process, role of natural product, ethnopharmacology. Pharmacology is a group of people, they evaluate the drugs efficacy scientifically. Ethnopharmacology means the scientific study of substances used medicinally, especially folk remedies by different ethnic or cultural groups. Particular region or particular culture, they use certain plant materials for curing certain ailments. That ethnobotanical knowledge can be taken for the scientific segment and to prove that subject is called as ethnopharmacology. Thereby the drug discovery process takes place. And coming to the application of subject pharmacognosy. Pharmacognosy is related to botany, ethnobotany, marine biology, microbiology, herbal medicines, phytochemistry, pharmacology, and pharmaceutics. So pharmacognosy is relied as subjects for other specializations subjects too. It's an interlinked discipline. And the pharmacognosy is also associated a field with some poisons and hallucinations plants for the productions of oral contraceptives, for the allergens and herbicides and insecticides. This subject is almost means also allied with those area. Let me talk about hallucinogen plant. Hallucinogen plant means it feels the persons of a well-being, physically and mentally, even he she not. So that hallucination state is called as a feeling of well-being. The veratrum is there, a drug. Opium is there, that means a ganja. These are the plants are having the hallucination property. And most of times the natural products helps us for the synthesis of oral contraceptives. And allergens can be obtained. These allergens means if we may be prone to certain allergic conditions in the summertime, in the winter time, there may be some due to some allergenic substances. It may be from the flower pollen grains, or maybe some dust, or maybe some insects. Those substances taken into consideration, and they prepare the extract out of that. They timely test with the patient. If it shows allergic responsibilities, hypersensitivity responsibilities, we can say you may be prone for this condition if you go and expose this area. So the allergens is a substances helps for the diagnosis purpose for the diseases. And what are the skills and roles of pharmacognosist? Because of their knowledge, their techniques, what they learned in their studies, and those skills can be applied in various sectors. So analysis of other commodities, so they might be absorbed, and the skills is required for testing the food, spices, gums, perfumes, fabrics, and cosmetics. And it can be, they are also used by the public analyst department of forensic sciences and the quality control scientist and the herbal industries. They also play a role in a pure sciences as an academician, or they can help for making a conceptual development, or they can play a role in the writing up the books related to botany, plant taxonomy, and phytochemistry. Botanist and chemist looking at the various aspects. And the same way, the pharmacognosy also be considered for their knowledge involvement. Scenario of pharmacognosy in past, present, and future. So many books are given in different aspects, but I've taken two classical examples as a case study to explain to you as well as for getting a proper understanding the natural product pharmacognosy. I mean the natural product, what happened in the past and present and future. Let me take one good example, quinine. I talked on yesterday, right? So quinine is a plant obtained from a cinchona bark. It is native to South American tree. which containing a rich of two major principles, chemical constituents, quinidine and quinine. Apart from chinquinidine and chinquinine, so many isomers are there. I'm majorly talking about the quinidine and quinine. You can see the left hand side top white color image, the two structures I proposed there. I kept there, sorry. And it was used by Incas as a dried bark, which is they mixed with wine and they consume. But it comes into use as a medicine in 1631. How many years back? Almost 400 years back. 1631 by the Rome people. And then people started extracting in 18th century, 1820, and it comes into a large scale of production in 1850, and people started synthesizing a different analogs of quinine base substrate in 1944 now. And so many stories are there what happened to 2000, 2005 and 2010. I'm talking about straight away to 2019. 2019, you know, pandemic situation. Yes, coronavirus, SARS-CoV-2, and for that ICMR, Indian Council of Medical Research, suggesting as a drug of choice for the treatment of corona patients by the modified structure of quinine. Quinine is a wonderful anti-malarial drug, and chloroquine is wonderful anti-malarial drug. Now you see the structure, one Cl group is there and top one hydroxy group is there. Because of the addition of one hydroxy group, it becomes a chlorohydroxyquinine. Now it plays a wonderful role to eradicate this coronavirus. It's available in the market in the form of tablet. It was existing in the market previously for the treatment of malaria. Now the drug reposition studies have been conducted, is now used in the market. So what past is, it was used by Rome, and middle of 19th century, presently in the 20th century, they started chemical synthesizing and producing this tablet. Now in future for the corona disease, we are going to use this also as one of the molecule for the treatment. That's a line about a natural product drug discovery and the scenario of the subject. And another example, another example is Texol, Pacific yew tree. It is called a Pacific yew tree, Texas brevifolia bark. I like to give another case example called as Taxol. It plays a wonderful role as an anti-cancer drug. It is called as Pacific yellow tree, called as Texas brevifolia. The portion of the plant used for getting this Taxol is bark portion. You can see the left hand side, intentionally I kept this structure, very complex structure. It is, we cannot be able to think to synthesize in the laboratory. Okay, so how the journey starts, and in present day also we are using a Taxol in the generic name of paclitaxel as an injection dosage of 300 milligram dose. In 1964, it is actively discovered by NCI, National Cancer Institution, and 66 it has been isolated in the form of generic product paclitaxel, and it was acting wonderful mitotic inhibitor and used for cancer chemotherapy, like a for the chemotherapy agent for lungs, ovarian, breast, head, neck cancers. Then the need becomes more, so we want more of Taxol compound. It's been tested, the isolated compound been tested, it acts as a chemotherapy agent for this kind of cancers. So in 1969, people started collecting widely, which is growing in that region, 1200 kg bugs, which gives only 28 kg of crude extract. Out of that, only one can get 10 gram of pure substances. And it has started treating with some patient. In 1975, they started to check what other cancers it may be useful and what other activities we can use for these substances. So they gone for many in vitro studies. For that, they required in 1977 almost 7,000 pounds bark requested to make 600 grams of pure Taxol. So 1 pound is equal to half kg, or maybe 469 gram, right? So that much of bark is involved for making a 600 gram. And then they checked in 1978, it is using, shows a mild active in leukemia mice. And the people have started asking, not asking, it's required more amounts of this bark. So again they started scratching from the wild sources 20,000 pounds, which is required for various animal studies. Then for after the animal studies, it comes to the human subject. There is a phase 1 to phase 4 trials are there. For the phase 1 trials, they request, it's required 12,000 pounds, and for phase 2 to go ahead. And for the phase 2 trials begins with the recognized of 60,000 pounds minimum needed, and the environmental concerns voiced. And everybody, you are scratching this much pounds of tree, this much of kilogram of tree, and one day it will not be available. So that was a questioning. And simultaneously in 1988, they found it's more effective for melanoma, and especially for the ovarian cases. Then the annual destruction started around 360,000 trees, 360,000 trees to treat all US cases. Then people thought one day it is not going to be more, and we need to find some solution for that. So some alternative pathway they determined. The alternative pathway determinations NCI cannot do, they handed over to the BMS, British Medical Science Department. They find some other alternative pathway for synthesizing the Taxol. In 1992, FDA has given approval for marketing 5 years copyrights, and they use the trademark called as Taxol. Generic name is called as Paclitaxel. Now anybody can use this Paclitaxel for the production. The royalty time is over. Then in 2000, 20 years back, the sales peaked like in the US and reached 1.6 millions. Now available as a generic. That's what I'm talking about. Paclitaxel injection, 300 milligram in a 50 ml injection vial. So they said what pathway? Different pathway. Of course, the starting material is from the plants, Texas brachyta, left hand side image what I'm talking, Texas brachyta. They undergo the extraction procedures and separation science by chromatography techniques and the crystallization process they go. They get an intermediate. That intermediate is called as 10-DAB, 10-D-acetyl bactacin. And after that, up to this stage, we require this plant material, and we are not going to destroy more. From that, this acts as an intermediate substance, and they go for the chemical laboratory synthesis. There are many huge steps are involved just to show. By that you may get so many related compounds along with the paclitaxel. So this intermediate gives rise another 4 to 5 compounds, including the paclitaxel. I want only the paclitaxel that possesses the good anti-cancer property. So again, one separation science involved called as chromatography, and people isolate a compound of paclitaxel that is available in the form of injection that we use for the treatment of many cancers till today. This is a journey of Taxol from 16th century to 21st century. And the question may come again, why do we need plants? These all importance are there. We are witness that using the medicinal products for many diseases to cure, and because of these are the reasons why we need plants. One, and still in a better way, one, it acts as a source of drug molecules. Medicinal plants act as a source of drug molecules. Most drugs can be synthesized. Still more economical to use the plant. And Papaver somniferum, time is going, no? Okay. Papaver opium is one of the plant containing a morphine, codeine, a strong medicinal pain drug still today. Ergot fungus is a medicinal plant, is a fungus type of plant grows in the rye plant, called as ergotamine, which is used for the drug of choice for headache, and ergometrin, direct action on uterine muscles, and a source of complex mixtures. This is a source of complex molecule that can be modified to medicinal compounds like steroids. and it acts as a source of toxic molecules. Plants also have some toxic substances. Plants also containing some toxic substances which can be used for the study the pharmacological uses, and also we can use those toxic substances for investigation of the mechanism of that particular drug, particular compounds. These all the possibilities are there if you study about the plant drugs or with the help of plant materials. And it also acts as a source of novel structures. Periwinkle is a Vinca rosea called as Catharanthus roseus, containing a chemical constant called as vincristine, is an alkaloid. People cannot think about this structure, such a complex structure, so it may be acts as a source of novel structures and a source of compound to use as a template for designing a new drugs. So from the Papaver somniferum, people can extract the opium alkaloid. From the opium alkaloid, one can isolate a pure compound called as morphine. Morphine, known to everyone, is a narcotic and analgesic drug. Is a no better painkiller drug is available till today. So once the structure worked out, if we wanted to improve that particular morphine, if we go by synthesizing, substituting replacing the hydroxy group, there are two hydroxy groups is there in the morphine structure. If we substitute with the acetyl group, it becomes diacetyl morphine, commonly called as heroin. It becomes an addictive drug. Then some modifications takes place in the hydroxy group by the methyl group called as methylated hydroxyphenolic compound. It reduces analgesic property and it becomes a wonderful drug for suppressing the cough. So codeine phosphate, codeine sulfate is there in the syrup formulation, still now we are consuming. And same way, if you convert in the form of dihydromorphine reduced to oxidized state by 2-acylation process, you get a potential analgesic drug. And it's as a source of plant drugs. As a powder or extract, we can use still in commercially proprietary medicines, as well as the Ayurvedic formulation available in the form of powder or in the form of decoction, or in the form of tinctures, or in the form of extractions only. The pure compound based, phytocompound based drugs also available like Taxol, like quinine sulfate, but still now the journey is going on, because there are certain complexities are there, because most of the active ingredients is unknown from the natural product. Active ingredient is unstable because you detached from the plant, and it will not be stable. If it is intact with the plant cell, it is stable. But for our benefit, for our purpose, we detect. So the stability issues may come. So it may be work, it may not be work, it may lead some adverse event. And isolation process is very difficult and it's too costly. Okay. And you will be surprised if I say almost 5 lakh species of higher plants are there on earth, and only 10% should be investigated. So there are huge potential is there in the plant kingdom. In future, we can access for the natural sources or the plant drugs based material for the intense screening of anti-cancers, antimicrobial, antiviral, anti-malarial in other segments also. So future, 80% of world population rely on natural remedies. So requirement, westernization of societies is required with the traditional knowledge. So westernization of society is required with the traditional knowledge, not only with the allopathic system of medicine. Extermination of species. So we are collecting from the wild sources, and one day it's going to be indistinct, endangered in situation. So alternative method we require to conserve those medicinal plants, and the taxonomies should be identified, and the gene mapping should be taken care and should be stored in the library. So which type of gene is present, so that helps to us to identify the species in future. So reading the gene pools is required. Natural resources are exhausted, which is already exhausted, so we need to go for some alternative system of production, one for the real cultivation, for the production of many plants material, and we can also go for the artificial propagation, what I mean the micro propagation, also called as tissue culture technique. We can bring out the new plants. So the conclusion of my second part of chapter is natural products is very important to the medicine, for the medicine. Exist in range of structures that one wouldn't think of synthesizing. So because of the complexity in the structure present in the plant material, people not able to think and people not able to produce. So we can depend upon the natural medicines which have more uses. Can act as a template for new drug development, for the new drug discovery. The lead molecule of extract can be leading to the newer molecule. So it's an untapped reservoir of new compounds. So far in the last two lectures, part 1 and part 2, I explained a lot about the definition and the history journey of pharmacognosy, and now I talked about the scope and opportunities and its applications and its various development phases, and I explained with some two cases also. This will gives you the wider idea about how to attempt the answer for the following questions which is asked repeatedly by the university. One is define pharmacognosy and write its scopes. You may anticipate 5 marks questions out of that, or maybe sometime 10 marks question. And another questions may come like write a note on history of pharmacognosy. The question they put in a simplified way, so you can touch as per your own convenient with the materials which I am providing in this presentation. So only thing, you should be very clever enough how to represent your answer. So if you do well and you get a good score. So thank you very much.